All right. So, Aditya Muthu Kumar, can you please mute yourself? All right, thank you. So, we'll start today's session. We'll look at few questions and like simultaneously discuss the concepts associated with them. Okay, I will start sharing my screen. All right. So, this tutorial session is kind of an extra session, which where I will try to discuss like 15-20 questions, uh, like other than what we have discussed since the last 12 tutorial sessions. Okay. So, which of the following statements regarding restriction enzymes and their use in recombinant DNA technology is incorrect? Okay. So type 2 restriction enzymes are most commonly used in cloning because they recognize specific palindromic sequences and cleave at a defined site within or near the recognition sequence without requiring ATP. Next is isoseisomers are restriction enzymes that recognize the same DNA sequence but may or may not cleave at the same position. Next is DNA ligase is used to cleave DNA molecules and produce sticky ends compatible with restriction fragments, facilitating ligation. And like the other option is natural function of restriction modification system, the RS function of the RSM, basically R and M systems in bacteria is to protect them from invasion by foreign DNA such as bacteriophage DNA. So out of all these options, which of the following is the incorrect one? This option C, DNA ligase, is used to actually ligate or join the uh, cleaved DNA fragments. Okay. So this option is incorrect here. Okay. So the correct option will be option C. And few things like isoseisomers are enzymes that recognize the same target sequence and cleave it in the same way. For example, SPH1 and BB1. Then there are neoschizomers, which are enzymes that recognize the same target sequence but cleave at different points. Okay, so SMA1 here is recognizing triple C, triple G, and XMA1 is also recognizing triple C, triple G. However, XMA1 is cleaving in between the first and the second C nucleotide here, whereas SMA1 is cleaving at the third and the fourth nucleotides in between C and G here. Okay, so these two, XMA1 and SMA1, will be called as neoschizomers. Then there's something called isocodamers, enzymes that produce the same nucleotide extensions even though they have different recognition sites. Like BAMH1 has a recognition site as GGATCC and SOU3A1 has uh, like recognition site XXGATC here. Okay, so it will cleave at this position, and here also when BAMH1 cleaves at this position, what happens here is it will generate sticky ends GATC. Same here, SOU3A1 will also generate sticky end, which will have an extension of GATC. Okay. Now, a researcher wants to clone a 1.5 kb gene into a 4 kb plasmid vector. Okay. So the gene of interest is 1.5 kb long. The carrier plasmid is 4 kb long. Now both the gene and the vector have a single EcoR1 site. The cloning strategy involves using EcoR1 to cut both the DNA strands and then ligating them. And after ligation, the mixture is transformed into bacteria, basically competent cells. Now, which of the following banding patterns would be observed on a gel electrophoresis if the cloning was successful, assuming the plasmid and insert are present in a 1:1 molar ratio and also including a control with religated empty vector. Okay, so in general, you have a 4 kb plasmid vector which have been cut with the restriction enzyme which has produced sticky ends. Okay, or it has been cut with the restriction enzyme, and your gene has also a gene of interest which is the insert here has also been cut with the restriction enzyme in question here. Okay, now the thing is, this plasmid vector will also have a high tendency to religate without the gene of insert, like without the insert being ligated there, and thereby forming the empty vector, or if the insert is there, it will form the recombinant vector. Okay, so out of these options, what might happen here? When you run the complete ligation mix on a agarose gel, what will like how many banding patterns would you see? First is if it's also they say that if cloning is successful. Okay, so would you see a 5.5 kb band? Would you see a religated vector lane 4 kb band and 1.5 kb band? Or in the successful cloning lane, you will see 4 kb band most intense and 5.5 kb band less intense. Or you will see religated vector lane 4 kb band single intense band, or in successful cloning lane 4 kb band less intense 5.5, so this will be more in, okay, 5 5.5 kb band less intense and 1.5 kb band intense. Okay, so here in general what happens is, now if you have, this is the 4 kb vector DNA, right? Now, and your insert is 1.5 kb long. Now, if this insert is getting ligated here in this vector, what will eventually you will have a 5.5 kb recombinant vector, right? So, however, this restriction digested vector can also religate, thereby forming an empty vector of only 4 kb size without the insert. Okay, so what will be the banding pattern on the lane? It will be actually the successful ligation of the 1.5 kb insert will produce 5.5 kb recombinant plasmid. However, what happens is, every time the ligation reaction is not really 100% efficient. So many empty vectors can religate back to their original empty, like without the insert 4 kb, having 4 kb size, and hence the resulting lane will contain a mixture of 4 kb empty vector and a 5.5 kb recombinant vector, right? And since the empty vector can religate more efficiently than the vector with an insert, the 4 kb band will be the most intense, and 5.5 kb band, which represents a successful ligation, but it might be less frequent ligation event in the particular reaction mix. Okay, so this is something when experimentally we perform, we tend to see these kinds of results as well. Okay, now two proteins, protein A and protein B, have the same apparent molecular weight on a standard SDS page gel. Now you perform a Western blot and find that your antibody for protein A recognizes both the proteins. Now, which of the following methods would be the most effective for differentiating the two proteins? Now, you have your antibody against protein A. However, it is recognizing both these proteins, and also uh, both these proteins runs at the same position on an SDS page gel. So how would you really uh, deduce that both the proteins are present in your sample or your protein sample that you have purified, like that? Okay, so the options given are: run the samples on native page gel and probe with antibodies, perform a second dimension 2D page separating by PI first, I mean the isoelectric point first, and then SDS page, Or would you use a Western blot with an antibody specific to protein A and another with an antibody specific to protein B? Or silence the expression of one protein using siRNA and rerun the SDS page or Western blot? So actually, both B and D are usable options here. However, 2D page itself, doing 2D page itself is a little more um, trickier thing. Okay. Uh, Even silencing a gene expression for a like protein expression, a gene which expresses this particular protein, that is also trickier. But these two, if you are here using antibody specific to protein A and another with specificity against protein B, then the antibody which is specific to protein A also is recognizing protein B. Okay, so it won't be very conclusive. And so an ideal scenario in this case would be either of the two methods, B or D, but this is more amenable where you silence the expression of a gene which encodes for suppose protein A or protein B. So thereby you remove its signal that will allow you to confirm the presence of the other protein. Option B is actually a valid method but is more complex than necessary for this particular type of question. Okay. So in general, what is 2D gel electrophoresis? Briefly, in the large population of proteins, when we isolate, suppose, a whole proteome from a particular cell extract or any kind of like from a particular organism, be it bacteria, yeast, or mammals, it might be possible that more than one protein has identical or close molecular mass in the as like how we saw in the previous question and may not be separated by page. It is also possible that more than one protein has a very close isoelectric point or pi. Isoelectric point is where the proteins have an like at the point where the proteins exist as um, like a Zwitter ion where the overall net charge is zero. Individual charge, it does have charged moieties, but the overall net charge on the amino acid or the overall protein is zero. So it does not really move in an electric field. Okay. And so it is possible that more than one protein has very close isoelectric point and separation is not really possible by isoelectric focusing alone. So many times combining these two methods can yield better separation. So what happens here is two-dimensional gel electrophoresis or 2D electrophoresis is widely used for analyzing complex protein mixtures where it separates the proteins in two steps. First dimension is isoelectric focusing, which separates the proteins according to their isoelectric points. Second dimension is SDS page, which separates proteins according to their molecular weights. And hence, the complex mixtures consisting of thousands of different proteins can be separated. So this is basically the first dimension where there are actually strips um, of like where you actually load your sample and let it run, and then this particular strip is taken, and instead of the stacking gel uh, like how, inside the same apparatus for that is used for SDS page electrophoresis, in the same apparatus you have this resolving gel, but instead of the stacking gel there you actually put this strip that was used for separation in the first dimension. That strip is actually placed on top horizontally here, and then you run the gel exactly as you run SDS page. Okay, so now the proteins from these strips, wherever it has been blotted here, that will actually slowly come into the resolving gel and then eventually separate out. Okay, and also the SDS used in the gel running buffer is sufficient to maintain the necessary negative charge on the proteins. Okay, first dimension proteins are separated based on the isoelectric point or pi values, and in the second dimension it's based on the molecular weight. And when we talk about siRNA gene silencing, what happens here is it refers to the inhibition of gene expression through various mechanisms, thereby preventing a particular gene from producing the protein it encodes. So siRNA gene silencing is achieved through RNA interference pathways, which is widely used in functional genomics to study the function of specific genes or to explore potential therapeutic targets. How does it really work? So what happens here is it works via RNA interference mechanism to direct this risk complex RNA inducible silencing complexes to recognize and degrade specific target mRNAs, and which is primarily or broadly broken into three steps. First is introduction of siRNAs. Exogenous siRNAs can enter the cell through transfection or viral vectors, and with optimized transfection reagents, the siRNAs are able to efficiently cross the cell membrane and enter the cytoplasm. Okay. And next, the siRNA can bind to these risk complexes. When siRNAs enter the cells, they bind to risk complexes and produce a single-stranded guide strand through unwinding. And the guide strand pairs with the target mRNA through base complementation. And finally, what happens is the target mRNA is degraded when the siRNA guide strand binds to the target mRNA. The risk complex cleaves the target mRNA, resulting in the mRNA being unable to be translated into proteins, hence achieving gene silencing. Okay. Next, a protein complex is described as a homodimer. Overall, like the homodimer is 120 kDa molecular weight in total, linked by non-covalent interactions with two additional non-covalent 30 kDa subunits. If this complex is analyzed on a 1D SDS page gel without a reducing agent, what would be the expected band profile? So if it's a homodimer, um, right? So this is a homodimer, 60 kDa, right? And these two subunits are actually, suppose they're interacting via some non-covalent interactions. Okay. Now, when there are other two subunits, 30 kDa each. Okay. Now, when this complete mixture is loaded on an SDS page, okay, SDS is a denaturing page, right? Now, when you are loading it here, the homodimer, which is having some non-covalent interactions between each of the subunits, will be separated. Okay, it won't run at 120 kDa, it will actually individually run at 60 kDa, and there are 230 kDa subunits which will actually run at 30 kDa molecular weight only. It's not that the homodimer will run as a homodimer at 120 kDa. Okay, because this is a denaturing page, the homodimer will actually run as 260 kDa, like there will be 260 kDa individual subunits which will run at uh, this molecular weight at 60 kDa only, and there are 230 kDa subunits. Because it is a denaturing page here, it will run at 30 kDa molecular weight, like at the level of 30 kDa molecular weight protein. If it would have been a native page, then this homodimer would have run at 120 kDa molecular weight. Okay. So out of the options, so what would be the expected band profile? Bands at 60 and 30 kDa molecular weight. Okay. Yeah. So SDS page separates based on the molecular weight of the constituent polypeptides. The homodimer breaks into 260 kDa subunits, and the 230 kDa subunits remain as separate entities, resulting in bands at 30 kDa and 60 kDa. Okay. So which of the following is the primary role of SDS in SDS page? What does real SDS page do? Option A is to neutralize the negative charge of the phosphate backbone of nucleic acids, to impart a uniform negative charge to proteins, making separation based on size, to cleave the disulfide bonds with proteins to separate subunits, to maintain the native structure of proteins for separation based on charge. Native structure of protein, this is a denaturant. It does not really maintain the native structure of proteins. This is a detergent. And it is a negative It won't be able to neutralize the negative charge. It itself is negatively charged. Okay, so basically what it does is it imparts a uniform negative charge to proteins, making separation based on size. Okay, so SDS is an anioning detergent that denatures proteins by disrupting the non-covalent bonds and coating them with a uniform negative charge, allowing separation by molecular weight. Okay, and we add reducing agents to reduce the disulfide bonds. Okay. Now, a mixture of proteins is separated using ion exchange chromatography, and the column is packed with a stationary phase containing DEAE groups, diethyl aminoethyl groups. The proteins are eluted using a salt gradient. So what will be the correct order of elution for the following proteins from this column at a neutral pH? So the isoelectric points of the proteins are given. So what do we have here? We have the matrix is what? The matrix is DEAE, which is actually an anion exchanger. Okay. So if something is an anion exchanger, that means the matrix itself is positively charged, which will bind to negatively charged constituents of the sample that is loaded onto the column. Okay. Now, the isoelectric points of all these proteins are given, and it has been said that the pH is neutral. That means if the pH is neutral, like pH is 7, then what happens is when the pH is low, that implies that the hydrogen ion concentrations are high, right? So, if this is an amino acid, okay, so this nitrogen is having a lone pair on it, right? So, if there's a higher concentration of H plus, this N can actually donate its lone pair and exist as NH3 plus. Okay. However, when the pH is high, this means H plus ion concentration is low, and that means this exists as NH2 and this exists as CO minus. So when the like overall the charge is negative, but again, I mean, definitely it depends on what is the R group here, but in general, the charge is negative here when the pH is higher, and because the H plus concentrations are lower, and when the pH is actually low, H plus concentration is higher, and overall there's a net positive charge on the amino acid. Okay. So what is happening here? The pH of the medium is pH 7, and your protein X has a pi of 9, right? So it is the pi is actually, the pH is actually lesser than the pi. So it will actually exist as a positively charged, it will have a positive charge on it. The matrix is also positively charged. This is also positively charged, so there will be repulsion. So this will elute out first. Okay. Next, you have protein Y, which has a pi of 6, which is again less than the pH of the medium. So it will elute out, but comparatively, um, this is also less than the pH. This is also less than the pH. However, comparatively, this will elute out first and this will elute out at the end. So overall, what will be the elution profile of these three proteins? It is X, Y, and Z. Okay. So DEAE, so correct option will be XYZ here, option A. So DEA is a weak anion exchanger with positively charged functional groups. It retains the negatively charged molecules, and at a neutral pH around 7, proteins with the pi below 7 will be negatively charged, while those with the pi above 7 will be positively charged. So when we talk about the protein with a pi value of 9, it will be positively charged and won't really bind to the positively charged matrix, which is DEAE in this case. So it will elute out first. and then protein Y will be slightly negatively charged and will bind to the column. However, the protein with the protein Z with pi value of 4.5 will be more strongly negatively charged and hence will tightly be retained on the column, on the matrix, will tightly interact with the matrix and elute out at the end. Okay. Now, which of the following modifications to a high performance liquid chromatography system would lead to an increase in peak resolution? Decreasing the particle size of the stationary phase. Uh, in Actually, there is a mistake here. This is another option. Increasing the column length, increasing the flow rate of the mobile phase, using a mobile phase with a lower viscosity. Maybe I should change it here. This is actually 2, this is 3. Okay. All right. So what are the combinations? Like which of the options are actually correct in increasing the peak resolution in an HPLC column? So if we are decreasing the particle size here of the matrix, so what will happen is um, smaller, the particles are smaller, it will be more uniform, which will actually improve the column efficiency and hence will actually increase the resolution. Resolution as in how much can it really set, how much is the distance, how much sharp and well separated are the two peaks. Okay. Like in the chromatogram. That is what resolution means, how nicely it is able to resolve between two different molecular weight constituents of a sample. If we are increasing the column length, what happens is actually a longer column provides more theoretical plates, which increases the separating power and improves the resolution. But if we are increasing the flow rate, what happens is many times increasing the flow rate leads to increased mass transfer resistance and decreased resolution, especially for larger molecules. Also, increased flow rate can also, what happens is if we are increasing the flow rate, if the flow rate is really high, the molecules won't really get like above an optimum flow rate range, specified flow rate range. If the molecules of your sample, constituents of your sample won't really get a good enough time to interact with the matrix. Okay. So that actually leads to a decreased resolution. Also, if we are using a mobile phase with a lower viscosity, it will actually improve mass transfer rates and reduce the pressure drop across the column. And both of these will eventually lead to improved efficiency and resolution. Okay. So the correct combinations will be 1, 2, and 4. Increasing flow rate actually uh, hampers the resolution. Now, which combination of stationary phase and mobile phase is characteristic of reversed phase high performance liquid chromatography RPHPLC? Okay. So usually it's reverse phase because the phases of the mobile phase and the stationary phase are reversed basically. Okay. So in general, what we have is the like in general what we in the normal chromatography that we looked at earlier, also in normal HPLC, what happens is the stationary phase is polar and mobile phase is non-polar. Okay, but in RP HPLC, stationary phase is non-polar. Most commonly, it's silica with having 18C residues or C8, like 8C residues, like all these long chains of carbon chains, hydrocarbon chains bound to it, and the mobile phase is polar, typically a mixture of water and methanol or acetonitrile. So this is actually the reverse or opposite of the traditional chromatography where the stationary phase is polar and the mobile phase is non-polar. And in RPHPLC, what happens is the non-polar analytes are retained longer while polar analytes elute out first, elute out of the column first. Okay. So the answer to this question, the correct answer will be non-polar stationary phase and polar mobile phase. Now, A microbiologist cultures E. coli in two different nutrient broths, medium A and medium B, and records the growth curves. Medium A contains glucose as the sole carbon source, while medium B contains both glucose and lactose. And both cultures are started with an inoculum of 10 to the power 3 cells per ml. The growth curve for medium B shows a biphasic pattern, dioxic growth. The medium B means which contains both glucose and lactose, with two distinct log phases separated by a short lag phase. In medium B, what really explains, what best explains the cellular events during the short lag phase that separates the two exponential phases. So options are: the bacteria have depleted the lactose and are now producing enzymes to metabolize glucose. The bacteria are in a period of nutrient starvation but have not yet begun the death phase. The expression of the lac operon is being induced while the genes for metabolizing glucose are repressed. Glucose has been exhausted and the bacteria are switching on the genes required for lactose metabolism. So, the ready to go source for energy is glucose. So, whenever any medium has both glucose and lactose, so bacteria tend to use, like E. coli, will tend to use up the glucose resource first, this energy resource first, and later when the glucose will get depleted, then it will use lactose. However, for using lactose, you need to turn on the enzymes that will actually metabolize, as in you need to synthesize the enzymes that will actually metabolize lactose. So there is a small lag phase, that short lag phase is there in between, that actually is being used by the bacterial cells to, you know, synthesize all the materials that are required, like all the substances, enzymes or factors required to use lactose as an energy source. And hence, that's why there's a short lag phase. And then again, uh, once it synthesizes all the required substances, enzymes, factors, etc., then it can use lactose as a energy source. Okay. So, what happens in dioxic growth is, correct option is D. The glucose has been exhausted and the bacteria are switching on the genes required for lactose metabolism. This is why there is a short lag phase in between. So, dioxic growth refers to the biphasic growth pattern seen when bacteria are provided with two different carbon sources. The bacteria first consume the preferred carbon source, which is glucose in this case, and once glucose is depleted, there is a short lag phase before the second and less preferred source, which is lactose. And during this intermediate lag phase, the bacteria express the genes necessary for the catabolism of the second carbon source. For example, the lac operon genes are turned on and expressed and then synthesized, like beta galactosidase, etc., are synthesized. The synthesis of the enzymes for lactose metabolism, such as beta galactosidase, is induced, thereby causing the temporary halt in cell division. Okay, so the correct answer is option D. Now, a bacterial cell culture is inoculated with 10 to the power 4 cells per ml into a fresh liquid medium. The generation time of the species is 30 minutes, and after a lag phase of 60 minutes, the bacteria enter the exponential or log phase. So what will be the cell density of the culture 150 minutes after inoculation? So here, what do we have in our hand? Um, we, yeah. So initial is 10 to the power 4, sorry, cells per ml. Generation time is what, 30 minutes. Lag phase is how long? 60 minutes. And so the question asked is, what will be the cell density of the culture 150 minutes after inoculation? So given time is 150 minutes. Not actually given time, so we can write it as The total time given is 150 minutes. Okay, so maybe T total. Okay, so if the lag phase is 60 minutes and it's the given time is 150 minutes, so what, so overall when the cell actually starts growing, the cells actually start growing will be, in general, the T will be 150 minus 60, which is 90 minutes. Okay, and if the generation time is 30 minutes, so number of generations n will be T by the generation time, which is 90 by 30, which is 3, right? And initial cell density, like number of cells, were 10 to the power 4. Okay, and so what will be the cell density or cell number will be, there's exponential division in the log phase, right? So initial is 10 to the power 4, 2 to the power number of generations 3. So 8 into 10 to the power 4 cells per ml. So option, sorry, what happened? The correct option is option B. 8 into 10 to the power 4 cells, this one, per ml. Okay, so basically we have to take care of the lag phase. We have to divide it, as in subtract it from the total time given, and then calculate the number of generations. And finally, um, what will be the cell density? We have to calculate using the number of generations and the initial number of cells given. Okay. Correct option is option B. Now, in which phase would you expect to observe the most endospores in a bacillus cell culture? Would it be the death phase, lag phase, log phase, or lag, log, and death phase? Sorry. Lag, log, and death phases would all have roughly the same number of endospores. Usually, endospore formation is something the bacterial cells do when under, during any unfavorable conditions or under any stress conditions or nutrient limiting conditions. Okay, so log phase, they are happily dividing. Lag phase is, it's a preparatory phase for the cells to really prepare for all the substances required for their division. Okay, and so the death phase is something when we would expect to observe most number of endospores in a bacterial cell culture. That is when, that is like during this time, the nutrient or the resources become highly limited. Okay, so it's not really favorable for the cells to grow. So the correct option is option A. Okay. A scientist is investigating the phosphorylation status of a protein. So they use a standard Western blot uh, procedure with two different primary antibodies. One antibody recognizes the total protein independent of the phosphorylation status, and another antibody specifically recognizes the protein when it is phosphorylated at a specific residue. So both the antibodies are raised in the same species. What is the most reliable strategy to analyze the phosphorylation status of the protein from a single sample? So, options are, you run two separate SDS-based gels and two Western blots, one for each antibody. Doesn't make sense because what will happen is, one for each antibody, one antibody will anyways, independent of phosphorylation, it will recognize the total protein. Okay. Then would you run a single SDS-based gel, transfer it to a membrane, and incubate the membrane with both the primary antibodies simultaneously? Again, one of them will definitely will recognize like irrespective of its phosphorylation status. You run an SDS-based gel, like you run a single SDS-based gel, transfer it to a membrane, incubate with the total protein antibody and detect, then strip the membrane and reprobe the same membrane with the phospho-specific antibody. Or would you, this is the option, or would you like really run a single SDS-based gel, transfer it to membrane, and use secondary antibodies with different fluorophores to detect both primary antibodies simultaneously. Again, this option also it suggests that the primary antibody has already bound to both the proteins. Now, one of the antibodies actually recognizes the proteins irrespective of the phosphorylation status, right? So that's why an ideal thing to do would be you run an SDS, single SDS page shell, uh, transfer it to a membrane, transfer the protein bands onto the membrane, and incubate with the total protein antibody first, which recognizes the protein independent of the phosphorylation status. And then you strip the membrane from strip the membrane of this particular total protein antibody. And then again, you add or reprobe the membrane with phospho-specific antibody, which will eventually go and bind to the specific uh, protein, which is phosphorylated at the uh, specific residue. Okay. So, option A, run two separate SDS page gels, two Western blots, one for each antibody. It is actually reliable, but this is more preferred. Why? Because what will also happen is if you're running two separate SDS page gels, eventually your loading amount or loading quantities of the uh, that you are loading there might be different. But if you are running a single SDS page and reprobing it again with two different antibodies after like with the like where you are stripping the membrane of the total and protein antibody and then reprobing it again with the second antibody, you are actually negating the issue of any uh, difference in the loading amounts of the sample. Okay. Or if there are any transfer inconsistencies during the two uh, separate Western blots, okay, during membrane transfer. So there might be different inconsistencies in general while running the gel or during transfer, during probing as well. So if there are any inconsistencies, those can be negated if we use this option C. Option B is unreliable because both the primary antibodies were raised in the same species. For example, any kind of whether rabbit, mouse, horse, goat, and secondary antibody, for example, anti-rabbit, would bind to both, making it impossible to distinguish between the total and phosphorylated protein signals. Option C is a classic and reliable approach, particularly when primary antibodies are from the same species. By stripping the membrane after the first round of detection, all the antibodies are removed, allowing a fresh reprobe with the second antibody. This ensures that the two signals from the two different sets of antibodies are clearly separated and comparable. And option D would only work if the two primary antibodies were raised in different species. As stated, both are from the same species. Okay. Next, Haemophilus influenzae must be grown on chocolate agar, which is blood agar, treated with heat to release growth factors in the medium. So, Haemophilus influenzae is described as what? So, in this scenario, it actually requires a special type of medium. This bacterium is like it needs special type of nutrient supply, nutrient resource for its growth. So this will be, this is a fastidious microbe here. Okay. So fastidious bacteria are defined as organisms that have complex or particular nutritional requirements. They can't really grow on standard laboratory media and require specific nutrients and environmental conditions to thrive. This fastidious nature means that they are often difficult to culture, which can actually complicate laboratory diagnostics and research. When we talk about the nutritional requirements, these bacteria may need specific nutrients such as vitamins, amino acids, or blood components, and some may require particular atmospheric conditions like elevated levels of carbon dioxide or specific temperature ranges. For example, Neisseria gonorrhoeae, a fastidious bacterium, requires enriched media like chocolate agar, which contains blood components for its growth. Okay. So, fastidious bacteria are clinically significant because their complex growth requirements can lead actually to false negative results in culture tests. This means that a negative culture result does not necessarily indicate the absence of the bacteria. They may simply be unable to grow on the media used for testing. Therefore, specific tests or alternative methods like testing for the nucleic presence of the nucleic acids specific to the bacterium might be necessary to detect these organisms because they won't really are not really that easy to culture for their specific like very specific nutritional requirements. So a research group, so the option, correct option will be option D, fastidious. Okay. A research group wants to prolong the stationary phase of a bacterial culture grown in a batch fermenter. So they propose four different strategies. Which of the following strategies is most likely to be effective in prolonging the stationary phase? Would decreasing the temperature of the fermenter to slow down the metabolic rate help, or increasing the initial concentration of the growth limiting nutrient help? Or one can introduce a continuous supply of fresh medium and remove waste products, or adding a bacteriostatic antibiotic to the culture once it enters the stationary phase. So in general, what happens in a batch fermenter is there's no fresh inoculum added, and the waste materials from the growing cells actually keep on accumulating. Okay. So if we really want to prolong the stationary phase, an ideal scenario would be to actually introduce a continuous supply of fresh medium and remove the waste products. Okay, so that they don't really die off. Like the number of cells don't really go down exponentially. They remain at the stationary phase. So what happens here is at the stationary phase of the growth curve for a bacterial cell, this phase is typically initiated by the depletion of an essential nutrient or accumulation of toxic waste products. Now, in batch culture, limitations is a batch culture is a closed system where no new nutrients are added and no waste is removed. Okay, this leads to the eventual entry into the stationary phase. Now, in a continuous culture, by continuously feeding fresh medium and removing the spent medium, a chemostat can maintain the bacterial culture in the log phase indefinitely, effectively preventing the stationary phase from occurring. This is a common strategy to maximize biomass production. Decreasing the temperature would actually extend the entire growth cycle but wouldn't specifically prolong the stationary phase. It would also decrease the overall growth rate because bacterial cells do need an optimum temperature for them to continuously divide. And increasing the initial amount of nutrients would extend the log phase and delay the onset of the stationary phase but wouldn't prevent it from eventually occurring. And adding a bacteriostatic antibiotic would inhibit the growth and could push the culture towards the death phase quite early rather than prolonging the stationary phase. Okay. So, what the question is asking, the correct answer would be option C. Now, a cloning experiment uses this particular vector PBR322, which contains resistance genes for both ampicillin and tetracycline. The foreign DNA is inserted at the BAMH1 site located within the tetracycline resistance gene. After transformation, the bacterial colonies are first plated on a medium containing ampicillin and then replica plated onto a medium containing tetracycline. Which colonies represent successful recombinants? So, what is happening here is, um, yeah. So, if this is your plasmid PBR322, so it has a gene sequence which uh, infers resistance to tetracycline antibiotic, and it also has a region which makes it resistant to ampicillin antibiotic. Okay. Now, in this particular tetracycline resistance, um, like the genetic sequence here, there's a BAM-H1 restriction site. Okay, BAM-H1 site is there, to which a gene of our interest has been inserted. So if any kind of gene is inserted, um, and gene is inserted, then there will be loss of tetracycline resistance, right? Because the functional product won't be formed. Loss of tetracycline resistance, and hence there will be no growth on tetracycline containing tetracycline containing medium, right? So now what happens is, if this recombinant vector has been put into competent cells, right? So all the cells which have taken up this plasmid will definitely be ampicillin resistant and will grow on ampicillin containing medium. Okay. However, because of the insertion of the gene here at the tetracycline resistance region, uh, it has lost its resistance to tetracycline antibiotic. Hence, the cells won't be able to grow. So what happens here is, first the transformed bacterial cells are plated on a medium containing ampicillin, and then replica plated. The same plate can be replica plated on another medium containing tetracycline. So there will be lots of colonies in general, which out of them all are ampicillin resistant, of course. But suppose when like after replica plating, what will happen is, suppose out of maybe these 10-12 colonies, only 5 are able to grow in sorry, in a tetracycline medium. So that means the ones which are uh, able to grow in a tetracycline medium, those didn't really take up, or those do not have the, not really take up, those do not have the gene of interest at this particular position. Okay, the ones which did not grow, suppose out of these, if these ones really grew on tetracycline medium, so these are not the colonies of our interest. These are the ones which are the colonies of our interest, which did not grow on tetracycline containing medium. Okay, because they're like, these cells have taken up the recombinant plasmid with the gene of insert in this particular restriction site. All right. So this overall thing is something which is called as um, this is called as insertional inactivation, right? So what will be the, which colonies will actually represent the successful recombinants? The colonies that will grow on the ampicillin plate but not on the tetracycline plate. Okay. Colonies that grow on both ampicillin and tetracycline plates are the ones which have taken up the vector definitely, or like it either have, it doesn't have the gene of insert at all, it has taken up the empty vector, or the gene of insert has ligated onto a totally different region, not at our specified region, the BAMH1 restriction site. Okay. Colonies that grow on the tetracycline plate but not the ampicillin plate, this is not the ones that we want for successful recombination. And colonies that do not grow on either plate, that means the cells weren't able to take up the vector at all. Okay. So selection on tetracycline plate, non-recombinants, cells containing the original non-recombinant PBr322 will be resistant to both the antibiotics and will grow on this plate. Recombinants are the cells containing the recombinant plasmid having the tetracycline resistance gene inactivated and are no longer resistant to tetracycline. These cells will not grow on the tetracycline plate. And after replica plating, by comparing the growth patterns on the two plates, one can identify the recombinant colonies. The recombinants will actually appear on the ampicillin plate, but will be missing from the corresponding location on the tetracycline plate. Okay. So now, in a molecular cloning experiment, a gene of interest is ligated into the MCS of a plasmid vector, multiple cloning site of a plasmid vector. The vector contains a functional LAGZ gene. The ligation mixture is used to transform competent E. coli cells. Now, after transformation, the cells are plated on an LB-agar medium containing ampicillin, XGAL, the substrate of beta-galactosidase and IPTG. Upon analysis, it is observed that a significant proportion of the blue colonies contain the recombinant plasmid, contradicting the expected outcome. The expected outcome is the recombinant plasmids, the recombinant colonies are white in color, whereas the cells which do not take up the recombinant plasmids are blue in color. But however, the observation is that a significant proportion of the blue colonies contain the recombinant plasmids, which contradicts the expected outcome. So, which of the following is a plausible explanation for this observation? The foreign DNA was inserted out of frame, leading to a premature stop codon that results in a truncated, non-functional beta-galactosidase fragment that still complements the host's LAGZ defect. The ligation failed completely and all colonies are derived from non-recombinant plasmids. The foreign gene has an internal promoter which drives the expression of a functional beta-galactosidase fragment even after insertional inactivation. Or the foreign DNA insert was successfully cloned but does not cause insertional inactivation because it is not within the LAGZ gene. So, option B, the ligation failed completely. This is completely not correct because we do see that recombinant plasmids are present. The analysis has shown that the recombinant plasmids are present. However, it is present in the blue colonies. So, the ligation didn't fail completely. Okay. And so if the foreign DNA was, it is not within the LAGZ gene. All right. So in general, when we talk about blue-white screening, what happens is this is your DNA of interest or gene of interest, and this is the plasmid vector. Okay. And this is the LAGZ region. All right. And this is the competent E cells, which codes for the truncated peptide, which complements the alpha peptide that is encoded by this gene stretch in the plasmid vector and hence forms a functional beta-galactosidase enzyme. Now there can be three scenarios when this foreign DNA is actually inserted within the LAGZ gene here, and hence if there's everything fine where there's no insertion, if this particular plasmid vector is introduced or transformed into these cells, E. coli cells, so overall the alpha and omega peptides will complement each other and form a functional beta galactosidase enzyme, which will eventually um, act on the substrate, um, which forms like XGAL or ONPG substrate, which will eventually be cleaved to form a like chromogenic product, okay, insoluble blue product. But if the foreign DNA is inserted in this particular region within the LAGZ region, then functional beta galactosidase won't be formed, and hence even after producing the substrate, only white colonies will be there. Okay, because functional beta galactosidase isn't getting expressed, and hence, as in, it doesn't really the complementation, alpha complementation doesn't occur, and hence this XGAL isn't getting broken down. Or it can happen that, so this is what is the ideal case scenario which we really want. But if in case the insert is there, the recombinant vector is formed, however the insert has been like is ligated it at a region outside the lag-Z gene construct, then the lag-Z stretch is actually intact, and it can actually encode for a functional alpha peptide which will complement the omega peptide, and functional beta galactosidase will form, and hence it will break down X-gal to form blue colonies here. Okay. Or one another case will be the foreign DNA hasn't really inserted, like hasn't really ligated at all. There's empty vector ligation which has religated. There's no insert, and then still the lag-Z stretch is intact. So functional beta galactosidase will form, and hence there will be blue colonies. Okay. So only the ones with insert, like ligation of the gene of insert in the lag-Z stretch, will lead to inactivated or non-functional beta galactosidase, leading to white colonies. But now the question that is being asked here is the so what we ideally expect is these white colonies are the ones with our recombinant vector. But somehow after the analysis, uh, like it was seen that the blue colonies had the recombinant plasmid. So what might be the issue here? So the issue might be, so the ideally recombinant should be white, non-recombinants should be blue. So in-frame insertions often produce a fusion protein which may be non-functional, but out-of-frame insertions generally result in a stop codon or very like severely affected or severely altered reading frame. So if the truncated product is still able to complement the host's defect, the colonies would be blue. This is a possibility. The second option, which suggested that the ligation failed completely, this is incorrect, because a significant proportion of the question says that a significant proportion of the blue colonies contained the recombinant plasmid. Again, the third option said that an internal promoter in the foreign DNA that we are actually inserting could drive the transcription of a downstream portion of the lag-Z gene, potentially leading to a functional beta-galactosidase product through alpha complementation with the host's delta M15 protein. Okay, this is a strong known phenomenon that is actually what many times in regular laboratory scenarios, experimental cases, uh, that actually happens, that can actually complicate the screening process. And another option, that was the last option, which suggested that if the insert is not within the lag-Z gene, it wouldn't be a recombinant detected by the screening method and all transformants would be blue. But this contradicts the premise completely that a significant proportion of the blue colonies are recombinants. Okay. So what the most correct answer will be option C actually. I should have mentioned it here. The foreign gene, whatever gene we have introduced, has an internal promoter which drives the expression of a functional beta galactosidase fragment even after insertional inactivation. Okay. Now, a 6 kb circular plasmid was digested with three different restriction enzymes, EcoR1, BAMH1, and HIN3. The following gel electrophoresis results were obtained. EcoR1 6 kb, BAMH1 6 kb, HIN3 is producing two fragments, and then a combination of these two are producing two fragments, these two are producing three fragments, and then these two are three fragments. So basically the question is asking us to construct a restriction map of the plasmid. Okay, so how do you really do that? So first, what the question says is, so what we have is, this is a circular plasmid. So EcoR1 is producing a fragment of only 6 kb. That means only at it has a single restriction site in this plasmid. Okay, because if only if this is a circular plasmid, where is it mentions that this is a circular plasmid. If there is a single cut, then only it will produce a linear strand, a single linear strand. However, if a particular enzyme has two restriction sites, okay, or two recognition sites, then there will be two fragments. right? Two restriction sites will give two fragments of the circular plasmid, whereas one restriction site will actually give a single like a single digested fragment. So here out of these we see that both BAM-H1 and EcoR1 are giving us uh, single fragment. So that means both have a single restriction site on the circular plasmid. All right. And in 3 we see that it gives two fragments. So this is actually what we can say that this is hin 3. Sorry, what is this is hin 3, which gives like one fragment as 3 kb fragment and another as 3 kb fragment. Okay. So now a combination of EcoR1 and BAM-H1 will give, because both have a single restriction site, it is giving only two fragments. However, because hin 3 has two restriction sites and EcoR1 a single restriction site, it is actually giving off it is actually giving three um, fragments. Okay, three different fragments of different sizes. Now, how do you really construct a restriction map? So first we should be starting with um, hint 3, okay, which gives off two fragments. Now let's suppose this is the circular plasmid in question. Let's suppose this is hint 3. It cuts, suppose this is like zero site in general, and it cuts off at another site because it has two restriction sites. It is cutting out at 3 kb. Okay, so this is 3 kb and this is 3 kb as well. Okay, now next you see when we see at EcoR1 plus hin 3. Now EcoR1 plus hin 3. Now we see that EcoR1 and hin 3 is producing a 1 kb fragment, a 2 kb fragment, a 3 kb fragment. Now hin 3 is cutting at two sides, right? So one 3 kb fragment is intact. All right. So then another 3 kb fragment is actually getting divided into one 1 kb fragment and another 2 kb fragment. So that means EcoR1 suppose is cutting at this particular portion. EcoR1, suppose this is the 1 kb region. So maybe I should change the color. So this is 1 kb stretch. This is 2 kb stretch. And this is 3 kb stretch. Okay. So EcoR1 plus hin 3 is giving us these three fragments. Now let's look at, let's change the color for BAM-H1 and hint 3. Okay. BAM-H1 plus hint 3. Now what is happening in this case is it is giving us again a 3 kb fragment, 2.5 kb fragment and 0.5 kb fragment. Now suppose we are placing here if we are placing a BAM-H1 site here, oh sorry, suppose we are placing BAM-H1 site here, which is a 0.5 kb fragment, okay, 0.5 kb. So that means this one till EcoR1 site is 0.5 kb. However, we see that when eco R1 and BAM H1 are used, there are no 0.5 kb fragments being produced here. That means this is not really the correct site. Okay, for BAM H1. Oh, sorry. Mm, okay, this is not really the area. So maybe with a different ink, should try out maybe with this. Because it's a 2.5 kb fragment with BAM H1 and 3.5 kb fragment. So this complete thing was 1 kb and 2 kb, right? So now if there's a 3.5 kb fragment here, so if we place the BAM H1 site now here at this position, there's a 3 kb fragment here and this is 0.5 kb. So this is also not really possible. So if we make a fresh map again, maybe which color to use? Maybe this one. So what is happening is hint 3 at 0 position, hint 3 at 3 kb, and you have eco R1 here at 1 kb. So HIN3 eco R1 and HIN3, this actually satisfies both these scenarios, both these double digestion scenarios. Sorry, not this one. Eco R1 HIN3 and the single digestion scenarios as well. But however, if we are placing BAM H1 here or in this position, it really, if we are placing BAM H1 here, it is satisfying the HIN3 and BAM H1 digestion scenario. However, it does not really satisfy the BAM H1 and eco R1 double digestion scenario. But if we are placing BAM H1 site somewhere here, this is 0.5 kb, this was 1 kb, this was 2 kb, right? And in between two HIN3 sites, there was 3 kb difference, right? So if now BAM H1 site is here at a distance of 0.5 kb, then now when there's no HIN3, so if um, only BAM H1 and eco R1 are being used. So if eco R1 is cutting at this position and BAM H1 is cutting at this position, so overall you have 2 plus this stretch plus this stretch, 2 plus 0.5 is equal to 2.5 kb fragment, and 1 kb here, and 3 minus 0.5, this is again 2.5 kb. 2.5 kb plus 1 kb is equal to, sorry, 3.5 kb. 3.5 kb fragment here, which actually satisfies the question where this double digestion condition of um, 2.5 kb and 3.5 kb fragments. So if we were to uh, not with the red, maybe with black only. So the final map will be HIN3, HIN3. It has two restriction sites, eco R1 at 1 kb, suppose this is 3 kb, and BAM H1 at this site, right? So this is 1 kb, this will be, this total, this half is 3 kb, so 1 kb, this remaining is 2 kb, this is 0.5 kb, and this total was 3 kb, 3 minus 0.5 is 2.5 kb. So now you see it, and also 2.5 plus 1 plus 0.5 plus 2. This adds up to 6 kb. Okay. So this will be the final restriction map of the given 6 kb circular plasmid. Okay. So actually, I thought the pen might not work. It was creating some issues. So I have written it down as well. Okay, so based on the same thing, whatever we discussed, it's written down here. So you can refer to the slides as well later. Okay. All right then. That's it for today's session. So thank you all for joining. I will stop presenting. And all the best for your exams. I mean, for this particular course. And thank you so much for joining. I will end the meeting for all.